Assalamu alaikum girls. This is your biology class and today we are going to study about genetic engineering, which is related to biotechnology because it is a field of biotechnology, like it's a branch of biotechnology. So genetic engineering or recombinant DNA technology involves the artificial synthesis, modification and the removal of repair and repair. You can modify the genetic material, which is a DNA. You can add and you can repair also. Genetic engineers developed in the mid 1970s when it became possible to cut the DNA. Now how do you cut the DNA? With the help of an enzyme, okay? As a result, the characteristics of the host organism could be changed. If the host organism is a microorganism such as bacterium, the transferred DNA is multiplied many times and it is possible to obtain millions of copies inside a bacterial cell. So for these millions of copies of the recombinant DNA, we use the host organism, and this is known as genetic engineering because we can modify it, we can remove the DNA and we can add the DNA of the host organism and we can repair the DNA of the host organism also. In certain diseases, we repair the DNA also. What are the objectives of DNA of genetic engineering? See, these are the objectives of genetic engineering. Please note down, it can come in your short answers. The isolation of a first of all is the isolation of a isolation which is of a particular gene or a part of gene for various purposes such as gene therapy. Next is the production of particular RNA. So isolation and then the second one is production. Isolation of particular gene, then the production of particular RNA and protein molecules. So production means whatever you need. For example, you modify, you repair and you add the DNA, then what happens? The production of a particular RNA happens and protein molecules. So you get the protein molecules because DNA synthesizes proteins and RNA. After that, improvement of the then first of all the production is taken place, then you do the improvement in the products of the enzymes, drugs, commercially or organic chemicals. Like after the production, you can judge and you can change the products more, okay? Production of varieties of plants having particular desirable characteristics. So uh, the plants can be changed, for example seeds, and even the plants and the fruit also having particular desirable characteristics. Treatment of genetic defects in higher organisms, like in humans, there if there is a disease which is genetic, you can treat the disease with the help of these biotechnological or techniques of genetic engineering. So what are the basic steps of of genetic engineering? The first is the isolation of gene interest. Then you have to isolate the gene. Then you insert the gene in the vector. Third step, transfer of the recombinant. Recombinant means the changed DNA. So you transfer the recombinant DNA into the host organism. Number 4 is the growth of the genetically modified organism. Number 5 is the expression of the gene. Okay, so we have to study about the steps. Number first is the in the first step, the genetic engineer identifies the gene of interest. So whatever we need we need from the donor, that gene is identified. So donor organism is like is going to be studied and then we identify the gene of interest. Okay, special enzymes, which we already know, restriction endonucleases, they are used to cut the identified gene. So please remember, in the first step, restriction endonucleases are used. The name is mentioned in the first step for cutting the gene of interest from the donor. A vector is selected for the transfer of the isolated gene of interest to the host cell. So now we need a vector. A vector is actually for transport. So we have to add the gene into the vector, and which is usually a plasmid, the extra chromosomal DNA, which is present in many bacteria and it has a small circular shape. The gene of interest is attached with the vector DNA by using endonucleases. So endonuclease, it's a breaking enzyme, and ligase. See, and nuclease means where there is the use of ACE. It means something breakdown of something is seen, and where there is ligase. Ligate means to join. So ligate means to join. So ligase is for joining the ligase is for joining these enzymes. The vector DNA and the attached gene of interest are collectively called the recombinant DNA that we already know. So please go through this process, and uh, you have to learn about the terminology and remember the words. Okay, remember the specific words. Next is the transfer of recombinant DNA into the host organism. How do you transfer it? Recombinant DNA is transferred to the host. In this way, the host is transformed into a genetically modified organism. That is, that we have to plant the vector inside the host organism. The GMO are provided suitable culture medium for growth. What is culture medium? Culture medium is the it's the food for bacteria. It's like a jelly-like substance which is of agar or something, like it's the food for bacteria. The bacteria grows on it, like it's present in small petri plates. So we grow the bacteria in petri plates, and in even in higher, if you talk about advanced higher growth, we use fermenters. Okay, so expression of the gene. Now the expression of the gene is seen. It contains the gene of interest and the manufacturers the desired product, and manufactures the desired product which is isolated from the culture medium. So in case of the different companies, they have very big tanks, and from there the desired product is collected. Now the achievements of the genetic engineering. You must know about the achievements of genetic engineering. That is, various achievements of genetic engineering are as follows. Human insulin gene was transferred into the bacteria. So the first is human insulin gene. This genetically modified bacteria became able to synthesize insulin, and because insulin was a protein which is synthesized only inside the body, so now diabetics are receiving this insulin, which is synthesized by bacteria, and the diagram is also shown in figure 17.7. So if they ask you about the cycle of like about the production of insulin, don't get confused. If they ask you about a diagrammatical representation of the production of insulin, you have to make this diagram, which we'll be discussing later, which is the full process. Okay, don't get confused. In 1977, an E. coli bacterium was created that was capable of synthesizing human growth hormone. So, in 1977, human growth hormone was synthesized with the help of genetic engineering techniques. The hormone thymosin is also proved effective against brain and lung cancer, and that is also produced with the help of genetic engineering. And beta endorphin. Beta endorphin is also produced with the help of genetic engineering. It is a painkiller, and genetic engineering also produces safe vaccine against the foot and mouth disease, which is a viral disease in goats and sheep and deer. Similarly, many vaccines have been produced against human diseases such as hepatitis. So these vaccines are also produced with the help of using the genetic engineering techniques, which can be any one of them, which can be the production of protein with the help of addition of certain addition of genes, repair of genes, and modification of the DNA. Interferons are the always remember interferons are the antiviral proteins which are produced by cells whenever there is viral infection. There are interferons which are produced inside the body, and now it can also be produced with the help of genetic engineering. The urokinase is also an enzyme, and C kinase, where there is ASE, it is an enzyme because it has a degrading properties. Okay, it has degrading properties. It is used to dissolve blood clots. It is also used by genetically modified microorganisms. Now it has become possible to modify the genes in the human egg cell that can lead to elimination of the inherited diseases, which is hemophilia. Genetic engineering techniques can also be used to cure blood diseases like thalassemia, sickle cell anemia, which result from defects in the single genes. Normal genes could be transferred into the bone marrow. This is also possible because of genetic engineering. Genetic engineers have developed plants that can fix nitrogen. So nitrogen fixing plants have also been developed with the help of genetic engineering because the genetic makeup of the plants has been changed and they need less fertilizers. This is the process with the help of which insulin is produced and genetic engineering can be seen. So this is the human cell. We have to take the gene. This is the piece of the chromosome which is taken, and that gene is identified and then isolated and then inserted into the plasmid. This plasmid is taken from the bacteria. So you have to use restriction endonucleases to cut, and then you have to insert this gene here inside the bacteria, and now it has the recombinant DNA. Okay, now as the recombinant DNA has been inserted, now you have a genetically modified bacterium, and uh, all of the bacteria, it has a genetically modified plasmid, sorry, genetically modified plasmid, and or recombinant DNA is present, and now reproduction in bacteria is seen, and as a result we have production of insulin. Okay,